You know, it's incredible. We're just down the street, and this is the first time I'm visiting this campus, and it is beautiful. So thank you for the invitation. I'm going to talk to you today about something, it's like for now, something completely different. Very, very different from the last talk, but I understand this is kind of the way that this conference is constructed, that the topics are all selected to be very, very different and to kind of stimulate this discussion. So this is going to take on a bit more of a translational slant. We'll start with some of the basic ideas about episodic memory, and then we'll get into some of the fundamental work that we've done in the area, and then kind of try to go back and forth with translational models. So we'll start just by some definitions. What do we think of as episodic memory? And when we think about this in the human context, we always think about memory for events, autobiographical things that happen, events that happen on a day-to-day basis. But to be able to study this in a cross-species way, we have to kind of operationally define it in a tangible way using units that we can try to understand across species. So typically we use units like what, where, and when. So a conjunction of what happened, where it happened, and when it happened, wherever you have that sort of trifecta, we can maybe label that as an event, and we can try to study it systematically in animal model and also in humans. Now, we know, of course, from a lot of literature that this kind of memory relies heavily on the medial temporal lobes. It's not only on the medial temporal lobes, but it's a significant contribution to the medial temporal lobes. And we also know, which is of particular interest to my research group, that it degrades to an extent as we get older, but it also degrades much more significantly in the context of Alzheimer's and various other dimensions, but Alzheimer's disease most prominently so because it is defined first and foremost by memory loss. Now, the way that we take to study episodic memory is a little bit different maybe from the majority of how the field evolved to think about phenomenology like recollection and familiarity and sort of autobiographical or autonoetic experience. We don't tend to use those terms often in my research group. We tend to try to reduce things to maybe a little bit more of an information processing approach, maybe a more computational approach, and think about the kinds of challenges that our memory system is faced with on a day-to-day basis. And this is a particular challenge that I want to share with you. It is the idea that memories are highly overlapping. And we know this to be true. In fact, we know that our lives are pretty boring in general, aside from days where you have a nice conference like this. Every day you wake up, you have the same breakfast. The news, you go to the same office, you talk to the same people, you park in the same lot, you do most of the same things, and you go back home to the same family, most of the time at least. Right. This is Southern California after all. But our lives are filled with redundancy. There's lots and lots of overlapping information, and yet our brains don't seem to have any challenge with keeping those separated from each other, keeping those apart from each other. Yeah.
Sorry, there are some words in the chat that they can't hear you.
Oh.
Maybe just try to move the microphone.
Could folks in the chat chime in and just tell us if this is better? Here's a challenge. I can't see the chat up here, but anyone else can. Maybe they can confirm.
Oh, it's now better.
Okay, all right. It's just a little bit of echo on my end here, but okay, all right. As long as everybody can hear it, okay, I'm happy. Thank you for that. So when we think about these overlapping experiences in everyday life, we know that our brains can keep them separated from each other because we don't walk out, hopefully, we don't walk out to the parking lot to the spot where we parked the car yesterday or the day before. We walk out to the spot where we parked it today, even though that experience has a lot overlapping with all those other experiences. So how does our brain keep those overlapping memories separated? And the answer is, well, there's an app for that, right? It's a computation referred to as pattern separation, and the contention here is that this is a necessary episodic memory computation that allows us to distinguish among these similar memories, and that it requires the hippocampus. Now, that's not to say it's only performed by the hippocampus. In fact, we'll talk about some evidence that it can go outside the hippocampus. But the hippocampus seems to be exquisitely suited for this kind of computation. Now, the way to think about pattern separation is that if you have two overlapping representations, say A and A prime, you can think of it as a way to be able to distinguish them on the output side into an A and a B, two separable representations. And the modeling on this has suggested for some time that it is a function of the hippocampus but it also has to do with the interaction of the hippocampus with the cortex. And early on, when memories are still being formed and they're highly dependent on creating those hippocampal cortical traces, ones that you see here, you can see that there's sort of an index at the level of the hippocampus and then all of the disparate and somewhat overlapping bits that are sitting in the cortex. And yet the hippocampus holds on to that distinct index that is separated across those representations. There's another process on the flip side of this called pattern completion, which is not exactly the opposite of pattern separation, but it is the process of being able to bring back a prior or existing representation given a partial or a degraded cue. So you can think of this as cued retrieval. Say you're on the bus and you see somebody's face, and you may not recall their name right away, but eventually you're able to kind of piece back together their name, their identity, last time you interacted with them. Everything is pattern completed back to that prior experience, where you were given just a partial cue, maybe just a glimpse of their face. And this also requires this hippocampal cortical exchange, being that the hippocampus holds onto that index. Activation of any of these smaller components in the cortex might activate the index, which in turn then reactivates the entire trace. So these are somewhat simple, rudimentary ideas. Some of the indexing ideas go back to the work of Tyler and DeSena and others. But the building on that came from the work of this gentleman here, David Marr. And in the 1970s, David Marr did something absolutely incredible. In addition to doing some beautiful modeling of the cerebellum and so on, did a modeling of what was termed the archicortex hippocampus. And he was first to suggest that the hippocampus has inside it architectural features to be able to implement those processes. And just by example, by the way, a lot of these ideas have been evolved over time, further developed and modeled. In fact, one of the inspirations for me to start working on some of these ideas was a paper by Jay and Bruce McNaughton and Randy O'Reilly in 1995, a seminal paper at the time that was titled Complementary Learning Systems, talking about how the hippocampus and the cortex interact to be able to support these computations. And one of the things that we learned about from looking at the structure of the hippocampus is that there's regions that are sort of attached to each other that have these exquisite architectural features that allow us to implement these computations. What I'm talking about is this particular region, the dentate gyrus, which has a pretty high density of tightly packed granule cells. These are very, very small cells that are very densely packed. In the rodent, there's about 1 million of them. And they receive input, again, in the rodent from about 250,000 pyramidal cells in the entorhinal cortex, a massive divergence of signal that comes into the dentate gyrus. It also has a very sparse firing code. So it takes a lot to make those cells tick. Anyone who's ever stuck electrodes in a dentate gyrus, you know that one of the ways you know you're in the dentate is everything goes silent. Very, very quiescent. Unlike other cortices, other patches of cortex that may be a little more loud. Now, downstream immediately of the dentate gyrus, there's a couple of regions of interest. One is the CA3. That's the key one that we want to talk about here. That has another architectural feature that's really important, and it's a massive recurrent collateral network. It's capable of producing an autoassociative signal. And you could think about this in terms of attractor dynamics, but there's a number of different ways that this can also be instantiated. So the CA3 is capable of pattern completion, and it sits immediately downstream of the dentate gyrus. So the idea is that the dentate gyrus is this thing that's capable of generating a pattern-separated representation. It can project that onto CA3 through this incredibly powerful Mossy fiber connection. But if the dentate gyrus doesn't have that pattern-separated representation, doesn't have that pattern separation signal to project onto CA3, the CA3 is left to its own devices, its recurrent collateral network, and it can clean up representations, say, coming in from the entorhinal cortex directly, and move the whole system much more towards pattern completion or retrieval in this case. So you have this dynamic balance, this dynamic interchange within the hippocampus that allows it to implement both separation and completion. I'm glossing over a lot of details and massively simplifying what are actually much more complicated models here. So if there are additional questions about how these models work, direct them all to Jay. I'm just kidding. I'm happy to answer them as well. But let me get to where this helps us in the translational case. So as we think about what happens to our brains as we get older, some of this stuff starts to make sense. So we know that as we get older, people will report, people, including myself, will report problems with things like episodic memory, so what we've just talked about, what, where, and when, particularly the contextual or source aspects of episodic memory. So you might remember what happened, but you have difficulty recalling where and when it happened and placing that exactly in time and space. You may also have difficulties with the recollective experience, so having those vivid recollections that we maybe used to have when we were younger, having those as we get older maybe becomes a little difficult. Contextual memory, again, a variance of source memory. And spatial memory and navigation are typically things that get compromised as we get older. And then finally, an increase in false memories, which often surprises people. I tell them as we get older, we tend to generate false memories with higher frequency. Interestingly, our confidence in those false memories also shoots up. So we generate a whole bunch of false memories that are completely illusory, but they're highly confident. So we give ourselves the illusion that we have this superb recall, but those details didn't actually happen. Now, the interesting thing about all of these things is that there's something in common, which is an impairment in pattern separation would give rise to all of these particular phenotypes, all of these complaints that we hear as we get older. But that's circumstantial evidence. We want to get a little more, dare I say, mechanistic, or dare I say, causal. I'm going to try to get there a little bit. We've got to do some experiments here so we can actually demonstrate this. We can't just say, well, it seems like older adults have trouble with this. Is there a proper impairment in pattern separation aging? Can we document it? And then, importantly, for causality, can we intervene? Can we change it? I'm taking Bill's definition, by the way. So we know from work from our colleagues, this is work by Michaela Gallagher and Howard Eichenbaum, the late Howard Eichenbaum, and Heike Daniela and others, showing that if you were to record from an aged CA3 cell versus a young CA3 place cell, you find that the HCA3 place cell exhibits a form of rigidity. It finds that if you were to look at how this cell represents the environments, for example, the circular environment versus the square environment, they're similar in many ways. They're similar cues. The young CA3 place cell has no problem at all distinguishing them. It has a very distinct representation here versus here. But the HCA3 place cell is promiscuous. It does not care in the slightest that there's a difference between those environments, represents this in the same way. So in some ways, it is rigid to that representational flexibility that's required. And this is a number of papers that showed this beautifully and reproduced it across a number of models. So now the question is, can we do something like this in humans? And we're not going to stick electrodes in humans and have them forage in it freely in an environment. That typically is not something IRB allows us to do. So what we do is we make the task, basically require them to do so. This is a very simple task. It's called a mnemonic discrimination task. We've termed it many, many different things. People tend to think of them as pattern separation tasks. And the idea here, the challenge, is that you're looking at images of everyday items, just coffee mugs, whatever it may be. And you're saying something very incidental about them. Either it's indoor, outdoor, big or small in a shoebox. It doesn't really matter what you say as long as you're orienting to the stimulus. And then later on during test, we ask you to do a recognition test where we show you some of the same exact items, some items that are brand new that you've never seen before, and some items that are similar but not identical, like this picnic basket, for example. Similar to this picnic basket, but is not the same one. And we're asking you to tell us if that's the same exact image that you saw before or not. Turns out this is really, really challenging. Not just for older adults, for everyone, but more challenging for older adults, and far more challenging for Alzheimer's patients. So what we discovered initially, this is many, many years ago now, is that older adults struggle disproportionately with this task. And we mapped it onto a form of representational rigidity that looked very much like what we see in the rodents. This is using high-resolution functional MRI. This is early work, going on 2 decades now with Craig Stark, looking at subfields of the hippocampus, dentate and CA3 regions in particular, and seeing a form of hyperactivity, which we also saw in the animal model, and an inability to shift the representation when items varied in their similarity. So we termed it a representational rigidity because that allowed us to kind of look at this across species. But then getting to the causality bit, what's really important is that we've tried to reverse this hyperactivity, both in animals and in humans, with success. So you can reverse it. It's a hyperactivity that does not reach the level of epilepsy, but it can be mitigated by a low dose of an anti-epileptic medication. You can do this in the animal model. You can do it in humans. You can reduce the hyperexcitability or hyperactivity and rescue memory deficits. So for the animals, it made them much more able to distinguish those environments. For humans, it made their performance on this task better. Okay? So far, so good? Am I going too fast? Okay. So now putting it all together, putting all those pieces together, this is some modeling and summarization work by my former graduate student, now assistant professor at UCLA, Stephanie Liao, who tried to kind of put the pieces of the puzzle together and try to uncover where we should go next. And I've explained already a couple of different facets. This hyperexcitability, I should say, biologically speaking, it is because, at least principally driven by the loss of inhibitory input in the hippocampus, which is a very well-documented phenomenon that happens in the aging brain. So when you have this loss of inhibition, you have a form of CA3 hyperactivity or hyperexcitability that, again, is modifiable. But one of the other things that we discovered is that it also is associated with loss of perform path fibers. And we devised the neuroimaging method to be able to look at that in humans and documented this phenomenon as well. So there's a loss of those input pathways coming into the entorhinal cortex to the hippocampus, which now prompts a very important question. What happens when you eliminate input coming into the hippocampus? Can you possibly expect the hippocampus to do a good job? And those of you who come from the computer science field are familiar with this adage, garbage in, garbage out. We had to make sure it wasn't garbage in. Otherwise, how can we possibly expect the hippocampus to do a good job? Which meant we had to venture into really uncomfortable territory for me as a hippocampologist who had the blinders on for many, many years, had to step outside of the hippocampus. And when we did that, it was magical. I'll tell you more about it. But it turns out that outside of the hippocampus, immediately outside of the hippocampus, the entorhinal cortex, which both projects to the hippocampus and brings in projections from the hippocampus, is not one size fits all. It's actually quite nuanced, and there's lots and lots of things going on. So I want to kind of tell you about the selectivity that we see in these regions. And it follows very directly from what and where kinds of pathways. Again, this is super simplified. This is based on work from Jim Kinnear and many others, showing that the pathways leading into the hippocampus are somewhat sort of distinguishable in terms of what and where contributions. So we know that the periorbital cortex and the lateral entorhinal cortex seem to be preferentially responsive to object-type properties, whereas the postrinal or parahippocampal cortex and the medial entorhinal cortex are preferentially responsive to spatial properties. And one of the pieces of evidence to kind of assure us that this is the case is if you look at some of the properties of the medial entorhinal cortex, for example, you see things like grid cells and other spatially tuned cells, which we don't typically see in the lateral entorhinal cortex. So there seems to be this dissociation, and it'll become very clear why this is important in a second. But the question is, can we look at this dissociation in humans? And the answer is yes. Again, this is work by a former student who's now an assistant professor at WashU St. Louis, just got his first R01, by the way, so very, very exciting. He took this pattern separation task that I described to you and created a variant that involved both an object pattern separation manipulation and a spatial pattern separation manipulation. And the spatial was just very simply metric displacement. Basically, whether the item shows up in the same exact location in space or whether it has moved on the screen. So you can either do object pattern separation or spatial pattern separation, a dissociation, maybe even a double dissociation. And what he tried to establish with beautiful behavioral work that took many, many months to optimize before he ever took into the scanner, and then with functional MRI, is that there are distinct processing streams within the medial temporal lobe for this task. The object trials involved a selective modulation in the lateral entorhinal cortex and perirhinal cortex, not so much medial and parahippocampal, and of course in the dentate and CA3, because campus loves everything pattern separation. And the exact opposite was true for the spatial trial. Now the modulations were medial entorhinal and parahippocampal, and once again dentate and CA3 because the hippocampus loves everything pattern separation. Doesn't matter if it's object or spatial. So here we have a double dissociation between those two pathways with this really interesting task where you can pit the two against each other. And we tried to kind of think a little bit about how this might manifest, how this might happen as information is coming in to the medial temporal lobe. And we sort of developed this idea that there's a hierarchical resolution of interference on the path to the hippocampus. So everything is overlapping as you're coming in from the ventral visual stream and dorsal visual stream, spatial and object information. Those are overlapping to an extent. You see this marked overlap here. But as you get into the medial temporal lobes, the parahippocampal and medial entorhinal on one side, perivinal and lateral entorhinal on the other side, you have a domain-specific, or at least domain-selective version of pattern separation. One in the spatial domain, one in the object domain. And as those two streams funnel into the hippocampus, which is now carrying out the ultimate sort of conjunctive or hyperdimensional type of representation, it is able to do this fully pristine version of pattern separation where these conjunctive representations are finally separated from one another. Okay? So that was the idea. That was sort of maybe the fundamental nuance behind it. But we were after this for a translational reason. We were after the entorhinal cortex because we suspected, in line with previous literature, that patches of this entorhinal cortex are going to be more vulnerable to aging and Alzheimer's than other patches. It goes back to the idea that the lateral entorhinal cortex may actually be ground zero for Alzheimer's disease. We know that the lateral entorhinal cortex in the transentorhinal region, which is what you're seeing here right around the rhinal sulcus, is one of the very first places to deposit tau pathology in Alzheimer's disease. It's what we call Brock stage 1. So it is a hot spot where you see some of the earliest evidence of neurodegenerative change. We also know from work by Gallagher and colleagues that even if you look in an animal model that doesn't get Alzheimer's disease but just undergoes aging, like an outbred rat, 24-month-old outbred rats are senior citizens. They don't have Alzheimer's disease. They don't have plaques and tangles, but yet they have loss of reelin and increase in phospho-tau, two major hallmarks of the aging process. And they find them first and foremost in the lateral entorhinal cortex. And then finally, there's some evidence that tau may spread transsynaptically from the lateral entorhinal cortex, medial, and then to the rest of the medial temporal lobe, and then to the rest of the brain. So there may be a seeding property to the tau that manifests early on in the lateral entorhinal cortex, which is really important from a disease perspective. If you suspect that this is the first place where this tau pathology may be manifesting, that may be where you want to intervene. That may be where you want to target. So the question is, can we use this to try to understand something about aging, right? So Zach's previous experiments only looked at this in young adults. Well, his next experiments looked at this in young and older adults. Did the same exact task, separated out the spatial trials from the object trials to make it a little bit easier, because when you have older adults, you don't want a lot of task switching. And discovered a couple things that are really important. One is that the deficit on the object trials was much higher than a deficit on the spatial trials. This is in line with our predictions. But two, and very importantly, the lateral entorhinal cortex exhibited this loss in activation in older adults that we did not see elsewhere in the entorhinal cortex. It was just the lateral entorhinal cortex, and only on the object trials. It was a super specific interaction, just in this part of the brain, just during this condition. No differences in the spatial condition, and no difference in the medial entorhinal cortex. And once again, he was able to independently reproduce our prior hyperactivation findings in the dentate gyrus and CA3. So we started to look at this as a network imbalance problem. Dentate and CA3 hyperactive, LEC hypoactive. And maybe that imbalance leads to sort of a deprivation of the hippocampus from its input coming in from the cortex and allows it to rely on its recurrent collateral network, which is hyperactive, to essentially fall into a pattern completion mode rather than pattern separation. So that's the nature of the computational bias that happens in the aging brain. You fall more into completion than separation, more into retrieval than learning new information, which is exactly what we see happen in older adults' memory loss in general. So when we look at it from a network standpoint, now we can develop all sorts of interesting measures, like a ratio measure between the dentate CA3 and the LEC. And when you do that, you can look at older adults and young adults and see that there's a big difference in this ratio measure. And importantly, this ratio measure also correlated with their performance on the task. So it was predictive as far as their behavior. one thing to think about is, can this ratio measure be potentially a biomarker in a clinical trial where we try to restore it? We change this ratio somehow, reduce the hyperactivation, and maybe rescue the lateral entorhinal cortical activity. And there's some hints that we can do that. Okay, so now skipping ahead a couple of years, I have now a brilliant, actually postdoc turned recently assistant research professor in the lab, Jen Adams, who's done beautiful work on connectivity in this network. Now she's looking at a much larger sample of deeply phenotyped, very well characterized older adults who all have risk factors for Alzheimer's disease. They have amyloid scans, tau scans, MRIs, all sorts of really interesting information that we can now associate with disease and pathology processes. And what she found was that the connectivity between the lateral entorhinal and dentate gyrus is increased. And by the way, functional connectivity increases can happen if both of them are going in the same direction or if they're going in opposite directions. As long as it's systematic, we get an increase in functional connectivity. So it's agnostic as to directionality. And she finds this in those who are low performers on this object pattern separation task compared to the high performers. But then importantly, associates this with their amyloid status. So if you look at their amyloid uptake measures, which is a hallmark Alzheimer's pathology, the higher this hyperconnectivity was, the higher the amyloid load for those individuals. Now, none of them are Alzheimer's patients, by the way. They are all cognitively intact older adults, but they are at risk of Alzheimer's disease by virtue of having amyloid. And almost all of the work that I'll show you does not have MCI or Alzheimer's patients. It has older adults that have all of the risk factors. So we're talking about changes that happen very early in the game, maybe 10, 15, even 20 years prior to symptom onset, but a large number of those individuals are going down the path to Alzheimer's disease. Okay? Well, one of the other things that we were able to launch in the lab recently was tau imaging. So now we have PET scans that allow us also to look at tau uptake in the brain. And that's really important because it does have a spatial distribution. So it's not simply, tau positive or tau negative. We want to be able to stage individuals based on where the tau is, given that it has this spatial property or spatial spread. This is some of the images that you see here come from a tracer called MK6240. This is a second-generation tracer. It's very powerful. It does very, very well with picking up where tau is and not having a lot of off-target binding effects. And what Jenna discovered was that if you were to look at uptake of this tau tracer, both in the hippocampus and in the entorhinal cortex, you see an inverse relationship with object pattern separation. So the lower the scores, the higher that you have, or the lower the performance, the higher that you have this tau in the brain. And if you were to look at this in spatial pattern separation performance, you don't see that effect at all. So in other words, again, it is tying this to the function of the piece of the entorhinal cortex that we think is impaired. The object pattern separation components, that's what seems to be associated with, or performance on those trials is associated with tauopathy, and not performance on the spatial trials. Again, very, very specific, very selective, and of course you don't see any evidence of this if you just look at standard neuropsychological assessments of memory. If you use delayed recall and a word list learning test, which is a classic hallmark test that we use for hippocampal function, you don't see these associations. So it's really about pairing that digital cognitive assessment that we've developed with the pathology, specifically in these regions in the brain. The direct correspondence. And then finally, to kind of tie it all together, another way to be able to look at this is microstructure in the entorhinal cortex and the hippocampus using diffusion imaging. And in this case, is using the diffusion imaging method that we developed. It is also very high resolution called Zoomit. And the way that this works is that you can look at measures like mean diffusivity, for example, and get an index of microstructure. In this case, the microstructural feature is cellularity. So on the one hand here, you can see low mean diffusivity usually means high cellularity, and high mean diffusivity usually means low cellularity. So this is an index of pathology. You can do this again at very high resolution within subfields of the hippocampus. And what she was able to document was a relationship between mean diffusivity, both in the entorhinal cortex and in the dentate gyrus, with MK6240 uptake in the entorhinal cortex. again suggesting that that increase in tau is tied to a loss in microstructure in the entorhinal cortex. And this is prior to any sort of volume loss or any frank neurodegeneration that you can pick up on standard clinical MRI scans. Right? This diffusion imaging approach is looking deep within the microstructure and telling us something about that microstructure that volume simply cannot. And then the last piece is if you try to associate it with performance on the object pattern separation task, you also see an inverse relationship, both with dentate median diffusivity and entorhinal cortical mean diffusivity. This is just hot off the presses papers under review. Okay, last piece of Jenna's work that I want to share with you, which is a brand new approach. This is a way to step out of the hippocampus, as uncomfortable as it may be, out of the medial temporal lobe even, and look at the entire brain, because it is a dynamical system, as we talked about before. And taking this dynamical systems approach, we can now take a look at brain state dynamics as they fluctuate moment to moment in time and ask, is there something about how the brain goes back and forth across these states that might confer a disease process or might be associated with bad outcomes? So the way this works is that we use resting state fMRI, or Jenna uses resting state fMRI, which allows us to image the brain just while the person is lying in the scanner at rest for about 10 minutes. She used the ADNI data set, which has a very high sampling rate, so it's what's called a TR of less than a second, so about 600 milliseconds, so it's a pretty rapid kind of sampling acquisition. And she's able to kind of create a regional activation space where these states are defined by co-activation of different parts of this network in this state space. The result of this is that you can now create a map that tells you exactly at what point in time every one of these states happen. So for every time series, we can have a moment-by-moment state allocation or state assignments according to those states that were extracted from the data. Give you an idea of what these states look like. Now, this is entirely data-driven. You can do this the only thing is you can do it with K-means clustering, which is what we used here. You can also do it with hidden Markov models. You can do it with a number of different approaches, but the K-means does a very good job. And the only thing that you need for a K-means clustering algorithm, as most of you know, is just setting the K, right? Setting the number of states that you want to see. And in this case, we tried a whole bunch of different variants. We tried a 4-state solution, a 6-state solution, a 12-state solution, and so on. Turns out the 6-state solution is the one that works best. That's the one you're seeing here. And you see things that you may be familiar with, like DMN. That's a default mode network. This is a somatomotor network. LIM is a limbic network. And then VIS is visual. And every time that we've looked at a different data set, the same 6 seem to pop up, whether it's young adults, older adults. So we think those are meaningful in many ways. But the question is there a particular state, or rather stickiness in a particular state, that may be associated with bad outcomes? And the answer is yes. If you were to assign people based on their persistence in a particular state more than another, so for example, if you look at individuals who have this kind of profile, which is a limbic profile, so they tend to be easier to get into the limbic state for them and harder to get out. They tend to spend more time in the limbic state, so what's called fractional occupancy is higher in that state. And persistence is another measure, or dwell time, whatever you want to call it, is higher in this state. When we look at those individuals, we find some really interesting things. One is that they tend to be older. They tend to be more of the folks who are assigned a diagnosis of mild cognitive impairment or Alzheimer's disease. They tend to be the ones who have a higher level of clinical impairment, and they tend to be the ones who have the worst memory scores. So clinically speaking, it makes sense that this, not the state itself is pathological, but persistence in this state at the cost of all the others seems to be pathological. And Jenna went on to also look at different levels of pathology because the ADNI data set does have those measures. So now she can look at entorhinal tau and sees a relationship between the degree of persistence in this state and entorhinal tau. She also sees this with temporal composite tau, so not just the entorhinal cortex, but including the entire temporal lobe. She sees this in the A-beta positive individuals versus the A-beta negative individuals, and in those with lower hippocampal volume. So when you look at all of these outcomes, this persistence in this limbic state seems to be associated with essentially all of the negative outcomes that we think are really important to study here. And again, I want to make sure it's clear It's not so much that the state itself is pathological. The limbic state is really important. It's a mnemonic state in many ways, right? Because the limbic lobe is what includes the hippocampus and other regions. So it's not like we don't want to have this state around, but its persistence or dwell in this state at the cost of the others. That seems to be the challenge here. So to summarize, a lot of the work that Jenna and Zach did in the lab, I kind of want to put it back in the perspective of biomarkers and the way that we use biomarkers to try to understand disease. And this is sort of the set of integrated hypotheses here, that the dentate gyrus hyperexcitability or dentate CA3 hyperexcitability is this condition that happens early and seems to confer some of the risks that we see later on, and it's modifiable. So if we can intervene early, that may be a good thing. A-beta, amyloid beta, to try to kind of tie it to some of the pathologies that we seem to understand reasonably well in Alzheimer's disease. Amyloid beta seems to have an indirect pathway to memory loss or pattern separation loss, and that seems to happen through some of the hyperconnectivity, hyperexcitability kinds of things that we're seeing in dentate and CA. Tau, on the other hand, seems to be directly associated with pattern separation performance. It doesn't require a lot of mediation, but it may still be doing this through the reduction in microstructural integrity of the entorhinal cortex. So one may be happening earlier than the other, and the logic of amyloid beta being tied to hyperexcitability and tau being tied to EC microstructure, sort of the idea that we're going after. Now, I told you before that we have had reasonably good results trying to reverse this hyperactivity, and that helped performance on this task. But again, getting back to causality, the ultimate proof or the ultimate piece of evidence that you really want to go after is if you were to use this anti-epileptic treatment or this reversal of hyperactivity treatment during the course of the disease, are you able to stave off cognitive decline? In other words, if folks are going down this path, are we able to bring it back up and stabilize it? Not just in a short period of time, but over a couple of years. That's exactly what this trial did. This is the Hope for MCI trial. That was the first trial of its kind to try to reverse this hyperactivity and longitudinally monitor the outcomes. So the trial was conducted by my colleagues at Johns Hopkins, Michaela Gallagher and Arnold Bacher and Marilyn Albert, and the results were just published last year, and I want to share with you what they. So this is a somewhat more substantial N. It's 164 participants. It's a phase 2B trial. And they all were selected to have a positive amyloid scan. So the idea is that you want to bring in patients with MCI who have a fairly high chance of converting to Alzheimer's disease within a couple of years. Okay? And when you're designing a clinical trial, you might want to do this because those are the patients that might stand to benefit the most. And they split them up, randomized them into two conditions. One is a placebo, and the other one is AGB-101, which is a low-dose formulation of an anti-epileptic known as levetiracetam. You might know it as Keppra. The drug has been off patent for many years, but they developed this low-dose formulation. When I say low dose, by the way, I mean an order of magnitude lower than what you might give to a patient with epilepsy. Okay? Because again, this is not seizures. This is a mild form of hyperexcitability. And the primary endpoint here wasn't performance on the task, and it wasn't hyperactivity per se. It was change in clinical and cognitive decline, a meaningful, FDA-approvable kind of measure that tells you something about quality of life in patients, something called the clinical dementia rating. In this case, it was the sum of boxes score, which captures across multiple cognitive domains and clinical domains. And the goal was to get to a 25% reduction in slope. If you can reduce the slope of decline on this measure by 25% over an 18-month period, that's a huge win. And when they first looked at the results, it was not that exciting. The overall mean change in CDR sum of boxes was about 17%, non-significant, fell short of what the primary outcome was supposed to get you. But one of the most interesting things that they did, and we're continuing to do now, was try to segment the groups. This was an a priori analysis, a pre-planned analysis. Segment the group by whether or not they were carriers of the ApoE4 risk gene. There was a lot of literature coming out suggesting that actually getting Alzheimer's disease with this risk gene on board versus getting it without this risk gene on board might actually come down to two different forms of Alzheimer's disease. And we had no idea which one would bear more efficacy. And here's what they found. ApoE4 carriers did not have an appreciable effect at all, about 10% change, negligible. But ApoE4 non-carriers had a 40% change in their cognitive decline slopes, far exceeding the 25% mark. So this now prompts a really, really important discussion, which is, well, what the heck is different about ApoE4 carriers versus non-carriers? We don't know. We have no idea. This is still pretty new. We're trying to grapple with it, trying to understand what that means as far as differential biology of Alzheimer's for ApoE4 carriers versus non-carriers. But the fact that the non-carriers responded to this treatment and the carriers didn't might suggest that as we start to look at amyloid or therapeutics down the line for Alzheimer's disease, especially with these approvals for anti-amyloid drugs and so on, we might need to think about Alzheimer's disease as how Alzheimer's disease is. Multiple different diseases with maybe multiple different kinds of treatments for different groups. A much more individualized precision medicine kind of approach. It looks a lot like what we've done with cancer, and it's a very uncomfortable space for Alzheimer's researchers to be in. So it's going to take some time to shift this paradigm, but that's what we're hoping to be able to do. Ultimately, the upshot is hippocampal hyperexcitability seems to be this targetable and modifiable condition. I hope that with this evidence, I convinced you that you can look at a causal way to be able to understand this, but I will withhold judgment on the mechanism, especially for the ApoE4 piece. So I cannot tie this to specific molecular cascades, specific neural mechanisms. We just don't have that information just yet. But I think that's exactly what we need to develop. So for now, it remains a promising therapeutic avenue. How are we doing on time, by the way? Excellent. I need exactly 5 minutes. So now I want to shift gears a little bit and tell you what we've pivoted towards in the last couple of years. As we started to develop a lot of this work with potentially modifiable kinds of conditions in the brain and trying to modify it with clinical interventions, it became clear, especially if we go down the route of there may be multiple different versions of this disease, it became clear to us in a lot of our work and many of our colleagues that there is one thing that seems to be undervalued and underestimated in the Alzheimer's research world, and that is vascular contributions to Alzheimer's disease. Now, I don't mean vascular contributions to vascular dementia or multi-infarct dementia or strokes or any of those things. Those are very different. We're talking about much more subtle cerebrovascular injury and dysfunction, CVID. It's a term that we have to coin to capture many of the things that we're talking about here. More subtle changes to vasculature that might predispose individuals to a higher risk of Alzheimer's disease. And when we do this, necessarily, for the sake of the science and also the moral responsibility behind it, we have to be able to do this in sufficiently diverse cohorts, which is something that the Alzheimer's disease field has struggled with for decades. So we know that Hispanic, Latino, and Black communities are at least 2 to 3 times more likely to develop Alzheimer's disease, and we know that vascular change, vascular dysfunction, is also much higher among those groups. And in this case, it's also associated with social and structural determinants of health, including bias, racism, stress, environmental pollution, exposure, all of those kinds of things converging to be able to create a really nasty situation when it comes to risk factors for Alzheimer's disease. So we pivoted our entire research program, which is what we call Beacon. Beacon stands for Biomarker Exploration in Aging, Cognition, and Neurodegeneration. Beacon is now renewed as a 2.0, so we just recently got a renewal from the NIH to be able to support this. And the entirety of Beacon 2.0 focuses specifically on these marginalized communities and the risk factors for Alzheimer's disease. We've done quite a bit building those community relationships, having a community advisory board, translating everything to Spanish, working directly with our colleagues that really know a lot more about this than we do to make sure that our approaches are effective, getting in the community, enrolling the right participants, and then making sure that this research truly benefits them. So very, very exciting kind of turn for us, and now the research group has increased in size and in dedication to this idea. And I want to kind of end on a little bit of the road ahead, what we're thinking in terms of the big picture, because it's not just Alzheimer's disease. So what I didn't talk about today is our work in neuropsychiatric illness, our work in early life adversity, and a number of different other areas that essentially borrow many of the themes that we talked about here, especially in terms of neuroimaging approaches and developing digital cognitive assessments like pattern separation tests. Now, this is really important because all of you know that brain disorders, brain conditions are a global challenge, affecting about a quarter of the world's population, a variety of different conditions. And yet, given this very, very high prevalence, if you look at their track record for clinical trials in CNS, so anything related to the brain, massive failures. 85 is being generous, actually. It's likely much closer to 90%. And there are many reasons for this. One, you'll resonate with this, we don't understand enough of the mechanisms. We don't have enough background fundamental knowledge of the disease circuitry, what these circuits actually end up doing in healthy, typical kind of circumstances and how they're impaired in disease. As a result, preclinical models just notoriously suck in Alzheimer's disease and in a variety of different cognitive disorders. Across the board, because we don't have that fundamental understanding really worked out, our ability to develop preclinical models is really impoverished. So translation from those preclinical models to the clinic remains very, very challenging. The joke, and it's actually a really, really sad joke in Alzheimer's disease, is that we've cured it 100 times over in a mouse. But we're a long way from curing it in humans. I hope maybe shorter now, but still. And there's also this unhealthy focus on pathology rather than circuitry. All about busting protein. We're trying to break down amyloid, trying to break down tau, and it's like, oh, those are maybe indicators of dysfunction, but that's not the real dysfunction. The dysfunction happens at the level of circuits and synapses. So we have to be able to address the disease at that level. And then finally, the cognitive outcomes that we use in clinical trials are really insensitive. I showed you, for example, instances where if you were to use standardized memory assessments, you see no change, nothing at all. But if you use some of the more computationally driven and very targeted assessments, like these pattern separation tests, that's where you see the action. And that's where you see very, very close relationships with circuitry. So our approach is this B2C approach, biomarkers and cognition. We develop sensitive cognitive tests. We pair them with biomarkers that are mechanistic. I hope I'm not in trouble for using that word. But the idea is to have biological proof of concept for any of these things. And then we can reduce the risk in pivotal trials. Because we can do the shorter, smaller trials initially, establish proof of mechanism before we go and invest hundreds of millions of dollars in large trials that are doomed to fail. One of the other things that we're doing now is, because it's in vogue, everyone wants to do it, but we really mean it, applying AI. So these tasks that I shared with you, they generate a wealth of information. It's not just accuracy. There's a lot of information buried in reaction time. We do various different models. We apply drift diffusion modeling. We apply a whole bunch of different models to it to try to extract constructs that are much closer to the cognitive processes. And we're now trying to use machine learning or AI to be able to develop a composite of these scores that is going to be maximally predictive. And we're doing this because things move a little bit too slow for comfort in academia. I've decided to also build something in industry. So I have one of my startups is Ognition Labs. It has a small SBIR that is funding some of this work in the laboratory. So full disclosure, there is a commercial interest here. But it seems to be the only way to really get things out to the clinic much faster. Academia, it just takes a bit longer. So that's the platform. The first phase was validation, and we've done a heck of a lot of that. The second phase, where we are now, is massive data sets. We're collecting data from hundreds of thousands of people across the world, not just across the country. And then the third phase, which we're hoping to kind of roll out in Q4 of this year, is starting to use this model training to be able to do some of the predictive diagnostics that we'd like to do. Okay, let me wrap up. So our take-home messages for today. I hope I've convinced you that you should care about pattern separation. It's this like really important thing that our brain does, and it requires the hippocampal dentate and CA3 regions, at least that sort of multidimensional version of it. And that object and spatial pattern separation seem to engage these distinct pathways within the medial temporal lobes. It may be a hierarchical processing, maybe, maybe not. But for the translational case, the lateral entorhinal cortex seems to be where a lot of the action is. That seems to be really important. It's tied to performance of the object pattern separation task, and it's tied to hippocampal and tau deposition, a real facet of Alzheimer's pathology. Hippocampal hyperexcitability, I hope I, again, showed you enough evidence to suggest that this is a modifiable condition, something that we can try to rescue early in Alzheimer's disease, perhaps with results down the line for a therapeutic avenue. And then one of the most interesting things that we're doing right now, or Jenna's doing right now, is these dynamic brain states analyses and identifying that there's sort of a stickiness to this limbic state that's associated with pathology. And one of the intriguing things that we're going to try to attempt in the next few months is, can we use an intervention and use this as a biomarker to see if we can get people out of this state? Can we make the state less sticky? Can we get people to cycle in and out of it a little bit more fluidly with ease? And then finally, cerebrovascular changes seem to be this early change marker and is associated with risk factors, particularly Hispanic, Latino, and Black communities. So it's associated with some of these disparities in Alzheimer's risk. And finally, combining biomarkers and digital cognitive assessments, I think, is going to be the way of the future, at least for us. But if you're interested in talking about any of this, I highly encourage you to come and chat with me about it. Finally, I want to just thank all the folks that do this work and allow me to come up here and take the credit for it. It takes a village. This is actually a small subset of our team from the recent Learning and Memory Conference of Huntington Beach. But they are incredibly hardworking and incredibly creative. One thing I should mention, that Beacon 2.0 study, that's NIH renewal, that was a massive renewal, and it was entirely crowdsourced by my lab. So they're the ones who wrote it. I just did a little polish. But it was all the science that my lab wanted to do, all this creativity, all of the things that they were really passionate about. And we were very fortunate. Got funded on the first try with a 3rd percentile, so we were just super stoked that this sort of team science idea actually worked when you put it to practice. And with that, I will stop here and happy to take your questions.
Thank you for that amazing talk. Lots of interesting stuff going on. I'm curious about the link between the anti-epileptic and its ability maybe to prevent the hyperactivity in the hippocampus. Now, I know that low doses of anti-epileptics are also prescribed as a prophylactic for migraines. And because you brought up the cardiovascular connection, I wonder if you are aware or have you explored the link between migraine prevalence and risk for Alzheimer's?
Wow. That is such a fascinating question. I have not at all, but now I have homework. So I think that's potentially a really, really interesting connection. I've got to do a little bit more digging to find if somebody's looked at this in the past and whether there's a link at the level of excitation inhibition imbalance, which is really kind of what we think is happening with hippocampal hyperexcitability. There's likely a link there.
Yeah, I mean, I'm wondering if there's data sets where you can look to see, like, people who currently have a diagnosis of MCI, whether they've had migraines in the past.
Yeah, I mean, I think we'd have to look and see if some of that data exists in the large consortium data like ADNI. We do ask about migraines and other symptoms in our Beacon study, but it's not a thorough assessment. It's one of many, many questions that we ask. And every now and then somebody will report it, but it's not, I wouldn't say it's high prevalence in our sample, but it's a small sample. We kind of focused on deep phenotyping in a small sample. ADNI is more thousands of people, so we could try to look at it there. Mechanistically speaking, I think it would be interesting to see whether, so in this case, the drug that I'm talking about, levetiracetam, it's an SV2A mechanism, right? Prevents glutamate vesicles from release, exactly. So there's other mechanisms one can think about, GABA agonists. There's a number of different approaches to go after it, but we tried to do this with a drug that already had kind of a great track record in the clinic and low side effects, and using it at that low dose also meant there's literally no side effects. So it may vary as a function of the anti-epileptic, what the mechanism
I think valproic acid is the one that's usually prescribed.
That's right. That's right. Yeah. Okay. Food for thought. No, definitely I'll look into it. Thank you for bringing that up. I appreciate it.
Totally amazing. This suggests to me that progress really is possible with science. just mind-blowing. My first question was going to be, do I need to be taking any of these drugs now?
Well, I've got some in my pants. I'm selling them out.
Two concrete questions. The first one, the first one's concrete. Is this association with minority status like separate, a risk that's independent of, or of that ApoE risk gene thing? I mean, is it like this is a cause and that's a different cause, or
Yeah.
how do you think about that relationship?
Yeah, that's a fantastic question. So there's a couple of different ways to think about it. One is when we're looking at minoritized groups, there's many different factors that play into this. And we have looked at genetics, for example. So ApoE4 is a risk gene, but it is not equally a risk gene across all ethnicities. Right? And I want to distinguish here what I'm talking about in terms of race, which is a social construct, versus ethnicity. So if we're talking about European ancestry, for example, versus African ancestry, there's a difference in terms of how much ApoE4 is a risk factor. It is less a risk factor in those of African ancestry. There are other genes, like ABCA7, that seem to carry more weight in those ethnicities compared to European ethnicities. So there are some genetic things that are interesting, but that's not the approach that we're taking here. The approach that we're taking here is to say there is a higher prevalence of particular risk factors, mostly that have to do with vascular change, that are also tied to a profile of adversity, of experience that those individuals are going through that is going to precipitate those kinds of deficits. So it may be that the profile of Alzheimer's disease that Hispanic, Latino, and Black communities are getting may be much more vascular in nature rather than another fill-in-the-blank nature for another group. So it goes back to the idea of there may be multiple forms of Alzheimer's disease, or maybe Alzheimer's disease is the end state, and there are many, many different pathways that lead to it. There may be, so when you think about cancers, for example, there's sort of a molecular phenotyping that you can do, or molecular subtyping that you can do. That's what I think we should be doing in Alzheimer's disease, trying to fingerprint individual groups based on risk factors and based on a particular set of mechanisms that might all converge on something in common, like tau and neurodegeneration. But it may be that tau and neurodegeneration here is driven by inflammatory and vascular differences versus something that is much more amyloid-based, for example, if we buy into the whole amyloid is pathological thing. Does that help answer it a little bit?
Yeah, so let me ask my second question, which is a bit more philosophical, I guess. You said fairly early in the talk that the propagation of, was it tau, is transsynaptic, or?
Possibly, yeah.
And, but that got me thinking about, okay, but something had to get it started in the first place.
Yeah.
And more generally, you know, if you had to speculate, because I know this has to be speculative at this point, about actual causal pathways that would get that going, I mean, what would you say? I guess that's
So let me
open-ended.
Yes, I love the opportunity always, Jay, to be able to just wave my hands and say a whole bunch of stuff and be on the record doing it. This is like my passion. This is what I love doing. So thank you for cornering me and allowing me to do that. But let me tell you what the field believes first, and then kind of where we might go from there. So tau is this interesting thing. So it's a microtubule-associated protein that exists. We all have it. Everybody has tau, and it undergoes a whole bunch of different conformational changes when it's phosphorylated. And if it's hyperphosphorylated, if it goes through too much phosphorylation, it falls off the microtubules, which allows the microtubules to disintegrate, and then that becomes a tangle. So the cell collapses upon itself. It can't shuttle things back and forth through microtubules. So in and of itself, it's there always. So the question about how does it transform, how does it become pathological, has to do with the type of tau protein and physical structure, and whether it's what's called sort of a seeding tau versus a non-seeding tau. So some of it seeds very quickly and is able to undergo those changes very quickly. And as a result of that, you're kind of seeing evidence of this tauopathy, or that change in structure and failure of the cell, which then causes tangles. But the question of transsynaptic spread is one that we don't have a good answer for. Some have hypothesized that it goes between synapses in the same way that prion diseases do. There's real sort of transsynaptic spread. But that idea is met with some enthusiasm from some, and some others are like, that doesn't make any sense. We don't see any real evidence for it. So whether or not it truly transsynaptically spreads is still contentious, but the fact that it starts at least being visible in the entorhinal cortex and then becomes visible elsewhere, that 100% we see evidence for. One of the ideas is that it is actually amyloid that liberates tau from the medial temporal lobe and allows it to go into the rest of the brain. But the entire sort of mechanistic cascade to get there is not very well worked out. I hope that helps. Yeah. Got more questions. Well, let's talk during the break. Yeah.
Okay, I'll have a quick one. At point 5 here on the summary slide, you mentioned the potentiality of using the sticky limb state as a biomarker for biofeedback. I'm sorry if I missed it in the presentation. What makes you think it is causal?
That's a great question. We don't know. We have no idea if it's causal or if it's a reverberation or an echo of something that happens upstream. But when we're after causal evidence, it was the same case with the hyperexcitability. The only way to know if it's causal is to try to reverse it and see what happens. So whether or not it's causal, the ultimate test is let's see if we can try to change it. And we can do this with stimulation protocols. We can do it with a number of different things that I think might be able to change it. But if we're able to do something to directly perturb network states this way, we're actually thinking about doing this also with neurophysiological recordings in patients with epilepsy. We can still do the same brain states analysis with EEG data. And if we can do this and see that there's sort of an impact downstream on the circuitry, then you can say it's causal, right? Mm-hmm. But without that evidence, right now, no, 100% correlation. I will own up to that. We still need to do that causal experiment for sure.
Mm-hmm. Thank you.
I know I'm holding you up for lunch, but Dr. Ross.
Really enjoyed the kind of nice full picture that included social structural causes and also, of course, a lot of mention of the circuit concept, which are interesting in so many ways. Some of the worries about the notion of mechanism are that it's often thought to be the kind of only or main type of causal structure in the world, but then it's also very reductive in a way that can't helpfully capture social structural causes or perhaps something at a mesoscale like a circuit.
Yeah.
A nice feature of having a clear, scientifically motivated account of causality is that you can show, hey, these are real causes, these social structural factors. Do you get any pushback with respect to those being legitimate causes and even something like the circuit, because it isn't the standard lower level mechanism at like the molecular level?
I think you're asking a really important question, Lauren, and I will tell you. So yes, there's pushback, but it's not what you might think. So I don't think anybody has ever pushed back on the idea that they are contributors. But there's been pushback as to say this is the cause. And I think there should be pushback to anything being the cause, because again, this is a multifactorial problem, right? So the pushback that we get typically is not so much that they are causal or that they are contributors or mechanistic contributors, whatever you want to call it. It is, but how are we going to do something about it? Like, how do we design the right studies with the right partnerships, with the right structure, so we can actually study this meaningfully, as opposed to just have a checkbox in our patient recruitment forms and say, we've recruited X number of those from these ethnicities. But you're doing the analyses without any real appreciation or understanding for those group differences. So when we've had discussions about this at the level of NIA, for example, and try to understand from those social scientists and others who have done a very good job of building those partnerships, the answer that we get from some colleagues in the community is, this seems really hard. This is like a really challenging problem. Duh, of course it is. Science is hard. But we have to do it. We have to start somewhere. So that's been the apprehension, is like, we're going to change the way that we do things. We're going to change the way these studies are structured, and then not have the simplistic kinds of models that we're used to be able to explain it. We're going to have to move into much more complicated territory, which is deeply uncomfortable. So, yeah, so I totally resonate with the question. Thank you.